I'm very happy to be here again in Oxford, so I'm most grateful for giving me this wonderful opportunity. One thing is to be here, and the other thing is to share with you our results. Most of them haven't been published yet, so I really hope that you will enjoy this talk as something maybe hopefully new. And at the very start, I'm not going to explain to you what the phages are, how important they are in the nature, or how we can treat infections with phages. I will jump to my major interest, which is how our body responds to phages. Because phages are stranger for human bodies, and the primary reaction of human body to foreign objects, this is the defense. And defense means the immune system. Immune system has traditionally been divided into non-specific parts. This is innate immunity, which is in the majority linked to inflammation. By tradition, we divide it into the innate part, which is mostly linked to inflammation, and to the adaptive specific part, when we have the specific recognition and specific response to individual objects. And this translates very strongly to immunological memory, which is really important in the reaction to the foreign objects. But what is very important, they do cooperate. This is the traditional understanding, but they cooperate and they cannot efficiently work without each other. And today I'm going to talk a little bit about these both parts. So we know that the majority of phage populations in our body, they are located in the gut. That's why the gut context, but I would like to show you how it extends to the whole body. Because of the gut, we chose endoscopic investigation because we are interested what's going on in mucosal surfaces. Why endoscopy? Not traditional, very common testing of feces, because feces, they may contain a lot of transient phages that come with food, water, and they not always reflect very well what's going on in the mucosal surfaces. And I will talk about two different studies here. One, it was a gastric biopsy, which means that we investigated stomach phages, and then we moved to intestinal phages. The first part, we wanted to see the cooperation between human genotype, bacterial microbiome, and phages. We invited volunteers who were donors of blood samples, and these blood samples were used to genotype and to look for specific SNPs that characterize the patients. In the same patients, gastric biopsies were used to investigate bacterial part of the microbiome and also the phage part of the microbiome. And as you see, there is a kind of triangle and the interactions of three parts, because of course, phages should not be analyzed all alone. They should be analyzed in the context of bacteria that are in the same individual. And what we found, the first thing what we observed was what we expected, that phages do correlate with their hosts. So it's a very natural expectation when we have a bacterial sensitive host, we expect to have phages that are in the same place, in the same ecological niche. However, we also observed that some phages were definitely present, but not really correlated to their bacterial hosts in the same people. This was a little bit unexpected, but it tells us that there are also other factors that definitely determine phage presence in human microbiomes, not necessarily if the bacteria are available there. So we investigated these SNPs. We investigated three basic groups. immunological factors, the genes that code for digestion, important proteins, and metabolic disorders, because we conclude that these three groups could be most important for what's going on in the gut and if the gut works properly. And interestingly, we observed that the majority of SNPs correlated anyhow to phage presence, to any phage groups. These were the immune-related factors. And here you have the list of the major ones. The bold ones were the most common. And as you see, we have here very widely affecting factors that very widely affect immune system. And for instance, interleukin-1 or TNF or pattern recognition receptors that are present here on the list, they demonstrate that phages, they are sensitive to the immune responses that our body can exert. Why this is important? Because this is what we usually think of how this looks like, phages in the gut. And this is what we actually have. I mean that all these tissues that may potentially enter the contact with phage, these are tissues really rich in immunological cells. And these cells are responsible for responding to microbiota, responsible for either innate or specific immune responses, and to a lot of regulation that are very well known to do on bacteria. But we know less, much less, how they behave in terms of phages. So because phages are here sensitive to genetic background that determines how these factors work, we can conclude that they most probably they do exert significant effects on the phage population, and they somehow affect the structure. Because what correlates here, this is the presence of specific phage groups that correlate to a specific SNP. Okay, and the same question as in the very general way. We have found SNPs that were linked to phages, but at the same time, they were linked to bacterial hosts. So for sure, we have this indirect regulation. This is a very well-known phenomenon that bacteria are regulated by immunological factors, but when we change the structure of bacterial microbiome, we do expect that phage community structure will be affected as well. However, the majority of these factors, they correlated to phages alone as well. So we maybe expect this peptidoglycan one. All others, they correlated to some specific phage groups without specific correlation to what we have in the bacteria. So we can conclude that we have this indirect regulation of phage, genotype, immune system, immune cells, and then bacteria that are affected, and this affects phage propagation and phage chance to be in the microbiome. But we also have indirect effects, like regulating just phages by themselves. And this brings us closer to the specific part and the specific elements that respond to phage. So again, phage population in the gut, and very well-known phenomenon that phages do translocate. They translocate, and this is the major way of phages to travel around the body, to distribute in the system. And in phage therapy, we hope that when we treat our patients orally, we get phages hopefully everywhere they will get to the site of infection, and we hope for their action. But what happens at the same time, these are anti-phage antibodies that are induced. The more efficiently, the more phage will be distributed around the body. Getting it more precisely, anti-phage antibodies, it's a simplification because antibodies, they don't target just the phage as it is. They do target phage proteins, and these phage proteins are considered phage antigens. As you know, they may differ from one phage to another, but there are also similarities. So it brings us to the idea that probably we have cross reactions. So analyzing phage targeting antibodies, it usually requires to identify what phage antigens are engaged in this reaction. And one step deeper, within the proteins, it's again not a reaction with the whole protein. These are small specific regions which are epitopes. Here just linear epitopes, but you may find other types. And these epitopes, this is what exactly enters the interaction with the antibody that recognizes the phage. So we can define amino acids that mediate this interaction between the phage and targeting antibodies. And how we investigated that? We used phage scan technology, and phage scan technology starts with downloading the whole database that we have on phages. So I'm very, very grateful to all people who work a lot on phage genomics, and they upload as much as they can to the databases, because then you can extract phage structural proteins. And these phage structural proteins, they are the key elements here. We take their sequences, we virtually cut into overlapping short amino acid sequences, oligopeptides that represent the whole phage proteome. Overlapping not to disrupt epitopes that could be found on the links. Then we translate it into DNA, synthesized by nucleotide printing. We have a huge pool of everything what we had in the database. Then we clone it in phage display system. And this is the key, because at this step, we have phages that contain both. In their DNA, they have this sequence coding for a specific oligopeptide, and the same phage particle displays this oligopeptide on the surface. And it's again a pool, a mix of very different ones. We use this pool, this library, expose it to human sera. In the serum, you have the whole variety of antibodies in a specific patient. We have the reaction, if only the reacting specific antibody exists. And then we use magnetic beads to get the fraction of these interacting ones, and we wash away everything what did not interact. And because of this coding sequence in the DNA, now we can amplify, sequence, and we have a pool of data on the epitopes that have been recognized in a specific patient, in a particular patient. And now we are getting back to our endoscopy department and what was done at this part of this work. So it was sequencing of phages that were present in the mucosal surface, again, the biopsy. And in the blood, we sequenced all the phage fraction that was circulating. And in the blood again, we used antibodies to see what specific epitopes were recognized in these patients, but their antibodies. So we had phageome in the mucosal surface. We had phageome that translocated and circulated in these people. And we had information on what phage epitopes were recognized in them. And what we got, it doesn't look very friendly, but here the details are not very important. What you have on the lists, these are always phage antigens that were recognized in specific fraction. But what is important here is that in the red, we have phage antigens that were found in the gut. In blue, we have phage antigens that were found in human sera. And in violet, we have the antigens that could be recognized in these patients by their IgG, the result of phage scan technology. And as you see, a huge amount of phage antigens in the gut, way less, but still detectable in the blood. But when we compare what was present in the blood, it's not what was recognized by these patients' IgGs, which clearly demonstrates how limiting factors are the antibodies that are induced by phages that somehow circulate or challenge by other way our immune system. And now a challenging try to summarize everything. So we start with immune cells that can be boosted by some immunological factors, and when they are activated, they are really responsive to phage antigens. These are primarily dendritic cells, macrophages, but also other cells that altogether work on recognition of foreign antigens. When phages translocate, typically they travel by lymphatic system, and it's very often that they can travel as the whole phage virus, but in many cases, phage antigens travel with dendritic cells, with macrophages, and they go to the lymph nodes. And within the lymph nodes, this is the place where immunological cells like lymphocytes, T cells, B cells, cooperate with dendritic cells and other antigen-presenting cells, and they learn how to recognize an antigen, and this applies to phage. Here we have dendritic cells that demonstrates phage antigens to T cells and B cells, because phages are in the majority consisting of proteins, which are typical antigens that are T-dependent. So we have a strong help from T cells, and this can result in real maturation of affinity, and we get strongly reacting IgGs and other isotypes of the antibodies. This may result in high levels of phage-specific antibodies in the blood, which means that phages typically are lost there. And because immunological cells, they travel everywhere. They don't stay in the lymph node. They go and locate in many tissues, including the gut, and they secrete more antibodies there, including IgG, IgA, which affects phages that are in the gut. And my take-home message for you is that we have immunity, including innate and adaptive immunity, that strongly shapes the phage fate in the gut, but also at the same time in the whole body. Thank you, and I would like to acknowledge my wonderful team who worked on this topic.